All right. I don't want to take any more of Eddie's time. We have a remarkable today. Look around the audience, everybody. Pretty amazing post-COVID. Okay. Thank you, everybody, for showing up. Of course, you would show up because we have a remarkable speaker this morning, and we're here to celebrate a legacy, Bob Goodkin. Now, for most of you in the room, you will not have worked with Bob Goodkin because he retired from the University of Washington in 2006 and died October 15th, 2012. And so Professor Chang is here to give the 10th annual Goodkin lecture. And the similarities between these two men are many, but the most important is their character. Bob was a consummate educator, and the residents knew him as an honest and honorable pawnbroker of academia. You could take whatever he said to the bank. He was that honest and that good. Ironically, Howard Goodkin, who helped me choose Eddie Chang as the 10th annual lecturer, knew that there was great overlap in their interests. Bob Goodkin was the first one to do deep brain stimulation here. He was the director of the Gamma Knife Center. And he was every time I think of Bob and all of us who knew him, he puts a smile on your face because he represented what's good in the world. And we miss him very much. And we thank the Goodkin family. Jared Goodkin's in the second row over here. Kind of an honorable, you have an honorary neurosurgeon of sorts. He understands a lot of the lingo because he grew up with his dad and he has to hang out with Howard. And Howard, thank you again for suggesting Eddie as our professor. Now, I could probably 30-minute introduction for Eddie Chang probably won't do it. So I'm going to try to focus on the high points that may be of interest to you. Edward Chang, of course, doesn't need any introduction. In 2020, he was inducted into the Academy of Medicine. That's sort of like going to the Hall of Fame in New York if you're a baseball player. And Eddie is the chair of neurological surgery at UCSF, arguably one of the, we say arguably one of the best programs in the country. We're very competitive, like Mitch is. There are a few things to know. Eddie, most important things that Eddie did besides all the academic, he's a father of 3 children, married to an anesthesiologist. He is first generation. His parents came here, grew up in Tahoe, and came here and worked hard. And Eddie got to go off to Amherst, where he graduated magna cum laude in chemistry. He'd go off to medical school at UCSF. He would be there for 2 extra years because he was a Howard Hughes fellow. And from 2010 to 2016, he had a meteoric rise from assistant professor to full professor. And 4 years later, he was to become the chair, and there was no one even close. He is that remarkable as a human being and as a scientist. In 2010, he co-founded and became the co-director of the Neural Engineering and Prosthesis Institute, which combines UC Berkeley and UCSF to train neural engineers and translate their research into clinical trials. So it's an amazing neural engineering center that Eddie helped build, and they're doing fascinating work. More about that. I told you that in 2020, he got elected to the National Academy of Medicine, which is amazing. He is the Joan and Sanford Weill Endowed Chair, and he runs the Weill Institute. at UCSF. Sandy Weil gave $100 million to do research. The University of Washington is part of that, but it's centered. The three institutions are UCSF, Berkeley, and UW. He is Howard Hughes Medical Institute faculty scholar. His awards take 2 or 3 pages, so I'm going to try to coalesce them. McKnight Memory and Cognitive Disorders Award. He won the Klingston Fellowship, and now he's on the committee, the National Research Service Award. He won for Klingston. He's the Wilder Penfield Award from CNS. Eddie has 9, let me repeat this, 9 active grants, okay? He has 2 UO1s, 2 program projects, that one is 3 million, one is 6 million, and they're really quite thematic. They're on the coding of speech and clinical trial. He's doing clinical trials for speech prostheses. He has 3 main research areas. The first one is to provide a mechanistic description for microelectrode arrays, real-time signal processing for the major properties and circuits that create speech and its disorders. And of course, his surgery, he operates while being chair and having 9 grants and having 3 children. He operates 2 days a week, sees clinic half a day a week, and is a productive neurosurgeon doing over 200 operations a year. I told you about CNEP, the Neural Engineering Institute. He also has a $30 million DARPA grant, which is to provide a mechanism. It's called Unlearning Systems and Neuropsychiatric Disorders. It is to understand brain circuits that go awry in neuropsychiatric disorders. Eddie has produced in his short career, remember he graduated in 2010, 292, almost 300 peer-reviewed papers. And they are not littering the literature. He has an H index of 91. That's like, Manny, that's like a passer rating, a football quarterback rating of 150, right? I mean, you can't even get that rating. So Eddie's simply remarkable. I met him when he was a medical student, and talk about Bob Goodkin personality. He was so honorable. He called me up. He didn't have to call me up. He said, listen, I really appreciate the tour and everything. I'm going to go to UCSF. And I said to Jeff, that guy's going to be something. Jeff Ochin, and I said, that guy's going to be something someday. I had no idea this. So without further ado, a person I respect and admire greatly, who certainly has many of the honorable characteristics of Bob Goodkin, and without further ado, Edward F. Chang from UCSF.
Well, I can't tell you how great it is to be here, and I need to take it down a couple of notches because that was just too much, and too much hyperbole. But I wouldn't be here for many reasons, actually, without this program. And life, I think, is very dynamic and interconnected. There's 2 reasons. One is the first time I took my boards, I failed. Just to put that out there, I'm going to take this down a notch. Took it again. Dr. Alan Bogan was a critical part of me getting through the second part. And, I'm grateful for that and his friendship. The second is that the legacy of this program, I think, weighs very heavily on my mind. The legacy of Dr. Ogerman, both Dr. Ogermans, George and Jeff, has been extremely important, informative in my own thinking and development, even the history going back to Dr. Ward. But really what's inspired me quite a bit when I was a medical student was the work that was going on here. And I think it's also no coincidence that one of my mentors, Dr. Berger, also trained and learned and honed his craft here in Seattle. So I'm here as an ambassador, as a friend, as a colleague from San Francisco. I hope that we can continue to develop a wonderful relationship. Had a wonderful weekend with Sam with digital neurosurgery down in the Bay Area. I think there's tremendous opportunities around AI for us to collaborate. And last thing, of course, most important is to thank the Goodkin family for this incredible lectureship and real privilege to be able to be here with you guys today. So one of the things that I'm going to really focus here is on the generation of the speech signal. What are the cortical electrical activity signals that are generated from the parts of the motor cortex that generate the words that I am saying? And it's a very complex signal, as you can see. There's been decades of work, including from Drs. Ogeman and others. And one of the places that we started in trying to understand this process actually even goes over 100 years ago to the time of Harvey Cushing. And many of you know this, but Harvey Cushing was an outstanding illustrator, in addition to being the father of modern neurosurgery. But one of my residents who rotated labs gave me this picture, which now hangs in my office. It's this picture that he illustrated where one of his patients had been shot with a bullet or something like that. The entry point was here in the forehead, and the exit was in the superior temporal gyrus. And this patient was suffering from intractable seizures. The thing that particularly interested me is what you can't really see that closely, but when I zoom into it, you see that there's this documented somatotopic organization from the face, the corner of the mouth, the lips, the jaw, the tongue, the palate, the vocal folds, moving from front to back in this very orderly representation. With the vocal folds all the way down here. So moving from the front of the mouth all the way to the back of the throat, moving from medial to lateral or superior to inferior. And this was in fact the precursor to Penfield's homunculus. So the homunculus, as Penfield described, was really the popularization of this representation, but it wasn't really the original discovery of it by any means. In fact, it was even known at this time. And I'm going to revisit, because this is the starting point for where we started looking at trying to understand how this part of the face is really important for speech representation. The homunculus that most of us are familiar with, of course, is this image, which is the popularization of that map. So one of the first things that we wanted to understand is how does that functional architecture, that map of the different parts of the vocal tract come alive as we speak? And the first way that we did this was using a technique called electrocorticography. Many of you know this, where you have implanted subdural electrode arrays for the localization of epilepsy focus, usually for 2 weeks. Now this is being replaced by stereo EEG, but occasionally we do have the reason to do electrode grids. And one of the first things that we did in my lab was to change from the standard 64 channel electrode to a 256 channel electrode that was 16 by 16 channels. So it really had the same safety profile of the lower density arrays, but allowed us to quadruple the information output that we could actually record and understand. And what allowed us to do was change the kind of questions that we were addressing, changing it from, is this part of the brain active during a given language task, to what's the nature of the coding within that particular area at each one of these electrode sites. So this is how it typically looks in our epilepsy monitoring unit, very small room, pretty crowded. Our patient here is engaged in a task. There are about 300 electrode wires coming out of the bandage. Here are two of our graduate students, Leah and David, who are doing a task, a postoperative fellow, and patient's mom watching on as we do these projects. And so what she's doing is looking at words on a screen. In front of her is actually a camera that's tracking the movements of her face, and a microphone as well that's recording the sounds from her voice. So like here, a lot of this work is done in collaboration with our patients and their families. We wouldn't be able to do any of this without it. And one of the reasons why this is so important is that these direct recordings from the brain are still by far the most highly resolved methods that we have to understand human brain activity. So fMRI is great for spatial resolution, but doesn't have the temporal resolution. It integrates over seconds. EEG has poor spatial resolution, but has millisecond resolution. These direct recordings have access to both millimeter and millisecond resolution. And that's what we need to understand processes like speech that are so dynamic, so fast, when individual phonetic elements themselves are only 20 milliseconds. So in this video, I'm going to show you some of the activations that occur in the sensory motor cortex. Here it's on the right hemisphere. They occur bilaterally when I'm speaking. And you'll see how quickly that information occurs in this part of the brain. And then it will be very apparent to you why we need these kind of methods and why I think over time they're going to become even more important to understand how this works. What you're going to notice is that it's not a blur of activity, but a very precise spatial and temporal code of electrical activity.
Shipbuilding is a most fascinating process.
So that flickering of activity is what we spent the last decade of my research career trying to understand. What is that code? Because that's the code that actually is controlling my vocal tract when I'm speaking. And what I mean by that is trying to understand what's happening at each one of these electrode positions. And then how do we bring it all together is something like an orchestra that gives rise to words. So one of the first experiments we did was to really do something quite simple, was to have the subjects read consonant syllables that were just 20 consonants with 3 vowels for a total of 60 syllables. It's the easiest possible experiment you could do. What we did was had them read this out. But it was really one of the first dynamic representations of this somatotopy that we'd ever had from speaking. And so just an example of some of the data that we found from this, if you look at the production of a ba sound, which requires the closure of the lips, and then the opening of that creates the sound ba. That's what we call the labial plosive. And this is the sound that's recorded from the microphone. And what you're looking at here is the electrode activity at dozens of electrodes, sort of from superior to inferior, as you're going across this real estate over the sensory motor cortex. And you can see that some of the electrodes aren't doing anything, but others are strongly activated. But one of the things that's most important here, as you see, is that it's not one particular electrode or one particular area that's clustered that gives rise to this sound, but it's actually something that is distributed, some here, some in the middle as well. And then if you compare that with a different sound, like the sound duh, when you make the sound duh, like in dad, the front of the tongue goes to the back of the teeth. That's called an alveolar plosive. And it's that closure of the vocal tract that gives the characteristic sound of a D. And if you look at the neural activity pattern for that particular sound, you can see some of it is very similar and overlapping, but other parts are very distinct. So this particular electrode is active here. This one is active here where it wasn't here. So now you're starting to see there's a distributed patterning going on, number one. It's not one particular location. And then number two is that it's specific to each speech sound. And if you compare it to a third sound, ga, where the movement that's required to make that is the back of the tongue going up, this is called a velar plosive. You can see that it has its own unique pattern as well. So what was interesting here was that it was not as simple as like one location as each speech sound. We had to basically try to understand what is giving rise to this particular pattern. So what we did was we created a vector matrix. And the simple way of just describing this was that for every consonant and vowel, from linguistics, we knew which part of the vocal tract is involved with making those sounds. For example, these labial sounds like ba, fa, ma, pa, va, wa. You can try sort of miming those yourself, and you see that these are all labial sounds. We created a matrix, and for all of those sounds, we were able to correlate the activity of certain electrodes in this particular spot of the sensory motor cortex. So this dotted line is essentially the central sulcus. What's in front of it is anterior. What's behind it is posterior. This is sorted from superior to inferior. And the electrodes that are in this part were the ones that were highly correlated with the lip movements, with labials. And then if you compare that with a different articulator, the larynx, we found something kind of surprising and something I didn't believe, frankly, at the time, which was we saw these very ventral localization of the larynx. Essentially, something had been described by many other people earlier, but an additional area that was really more in the precentral gyrus for the larynx. So, in fact, two areas were representing the larynx, and that had not really been shown before and was very surprising, which is why I didn't believe it. In fact, I did a very deep analysis of all the prior literature, including Penfield and previous to that, about our understanding. It just didn't fit. Why would we see something here in the somatotopic location, and why would we have two of these areas? So when you put it all together, basically, there is this laryngeal area here, a lip, the jaw, the tongue, and another laryngeal. And it's not as orderly and neat as I think we try to think about it, but that structure and sequence is there. And that's something that has been reproduced now several times by our group and others. So we then got very interested in trying to understand what is this separate laryngeal cortex. So one of my graduate students actually did his PhD on just trying to understand how the pitch of the voice is controlled by this laryngeal cortex. And his name is Ben Dichter. And what we're really trying to understand is how the brain controls the tension of the vocal folds to give rise to the change in pitch. So what Ben did was he found this task on Reddit. It's like, I really want to understand how we control this thing called intonation, which is a part of prosody. So he found this task where people say this sentence, I never said you stole my money. And what you do is you vary which word you stress. So for example, I never said she stole my money. It's just emphasizing what does it mean. So in English, we use that pitch to essentially convey meaning. And then if I change the pitch on the last word, I never said she stole my money. Same words, but really different, turns it from a statement to a question. So we use pitch to convey meaning constantly. And one of the things that Ben discovered was that this dorsal laryngeal motor cortex, which is really in the middle of the precentral gyrus, is where we have a lot of electrodes that control the pitch of the voice. And again, here you're looking at the right hemisphere. It occurs on both sides. These are the single trial pitch tracings from those trials where the first word was emphasized. So this is what's recorded by the microphone, the pitch tracing. And then below that, you're seeing the single trial electrode activity at this one particular electrode. You can see that the electrode is really strongly responding at this time point. But when you look at the trials where the second word was emphasized and the third and the fourth, it's tracking the pitch of the voice. It's not selective to the consonants and vowels. It's selective to those words that were stressed and where the pitch was actually increased. And it wasn't just this one electrode. It was a bunch more electrodes that were all localized to this area. So this was more evidence that there's a part of the brain here that is really about the laryngeal motor cortex, and it's not down here where we thought it was. And then when we looked even closer, we noticed that there is a small sulcus actually that divides the precentral gyrus, and it's usually located right beneath that spot. The arm representations in hand are right above this. So it seems like there is this dividing line between the different parts of the motor cortex, between what we call cortical spinal, and then below would be more cortical bulbar. One of the things that Ben discovered was that it's not specific to speech. In fact, when people were singing melodies, that this happens. And then also, it also has auditory responses there. So when we played back the speech that people, were that we recorded, we found that these areas were also responding to what they heard and other things as well. So it's both an auditory area and a motor area. And to prove that it was motor, not just auditory, because there's a potential thing that maybe this is all sensory, we wanted to prove causally, definitively, that this was in fact a motor cortex. So what we did was we took these endotracheal tubes called a NIMS tube, and our colleagues in OGNS use this for neck surgeries. So this endotracheal tube just has two wires along the ET tube, and they're placed really right down here at the level of the vocal folds. So you can actually do mapping of the larynx using direct cortical mapping. And we found many sites here that created EMG activity. So we proved that stimulation in this part of the brain actually can cause and evoke movements in the larynx. In fact, as you increase the magnitude of the stimulation, the amplitude of the EMG increases monotonically. Okay, so this is direct evidence that there's a causal relationship. It's not just a sensory auditory area, but it's directly a motor one that is causing muscle movements in the larynx. And we're still trying to understand why this is the case, why there would be a larynx area. But there have been several new papers that have described, this is not necessarily our idea, but people have recognized it, reproduced it with imaging, and now are speculating that this second laryngeal area actually may be an important part of the picture in the evolution of speech, because it has not been described in non-human primate. And I think that's to be determined, whether that's really the case or not. But this review from Eric Jarvis basically takes our findings and speculates that this area basically spread off from the primary laryngeal cortex to give rise to vocal learning. Okay, so we became very interested in this because anytime I see something that I don't understand, first I don't believe it, then try to get more data, see if it's really real. This was something that we kept seeing, more and more in the data, and we validated it using a lot of other methods as well. But what we're realizing is that this part of the precentral gyrus is actually quite interesting. And maybe we don't really think about it as just a motor cortex anymore. So I've described here that it's got direct motor control of the larynx. When patients are awake, and if you stimulate in this area, you can induce involuntary vocalization. There's auditory processing in this particular part of the brain. Some papers that are still under work right now, we've demonstrated that there's phonological sequencing and planning that's going on in these areas. And then I'll show you an example where lesions near here can cause apraxia of speech. So some of the key landmarks for those of you who are looking for this is that between the hand and this laryngeal area, there's a small cleft here. So we just call it a transverse sulcus, and does have an anatomical correlate to that. Something I never really appreciated until I just was looking at these images over and over again. So this is an example of an injury near that particular area involving it and someone who had developed apraxia of speech. This is actually a video from a colleague of mine who operated on this patient who had a recurrent tumor. And he's got normal language. He's got perfectly good normal language, and he's got full movement of the lips, jaw, tongue, the vocal tract muscles, but he can't speak. He's got a sequencing problem, which we call apraxia of speech. I'll just show you what that looks like. So very frustrating. He has normal language, can understand everything, can type, etc., but has, extraordinary difficulty in apraxia of speech production. And this is not an area that we canonically think of as speech. Right? This is like, if you look at this is not Broca's or Wernicke's area. And one of the ideas that we're advancing is that this mid precentral area actually is a critical area for the fluent articulation of speech. In addition to the corollary data that many of you in experience, many of you have here, which is that Broca's area itself is not so critical for speech production. It's really important for naming and higher order language processing, but not really for articulation of speech in the way that Broca envisioned. And so I think we're learning very quickly even about how this thing is all organized is very different. Another area that we've been interested in is not just the larynx, but understanding how the oral articulators are controlled by this electrical activity, not just the larynx. And this is a very, very understudied area in neuroscience. Like 99% of motor neuroscience focuses on the movements of the hand, the arm, limb, that kind of thing, right? Because you can measure it. It's simpler. It's like joint based. The orbicularis oris is a sphincter. The tongue has the dynamics of a hydrostat with dozens of muscles in it. Very, very, very complex. And on top of it, most of them you can't even see because they're inside the mouth. So we developed some methods to image using ultrasound and camera machine vision to track these movements. And I'm just going to summarize very briefly here that we've basically been able to identify, sort of how each position of the sensory motor cortex relates to all of the different movements of the vocal tract. So this is a mid-sagittal view of the vocal tract, and time is represented by the thickness of this line. And this is what we call the tuning or the trajectory encoding at a single electrode. This one particular electrode encodes a movement where the tongue tip moves to the front and then back down. You can see it goes from thin to thick. So that movement is in that direction. The tongue back goes down. So the basic projection of the tongue is from here forward. The larynx opens and closes. And then also this lower tooth, the lower incisor goes up and down. So this is a coordinated, not single articulate, but a coordinated movement. Tongue goes forward, the lower incisor goes up and down, even some movement of the vocal fold. All of this is encoded by this one particular electrode. And the reason I'm bringing this up is that we can't think of these areas as simply as just like representing necessarily like the simple muscles, but really thinking about how they are coordinating distributed movement. Okay, not just like thinking about it's like controlling this muscle, but they're controlling coordinated movements, and they tend to have these back profiles. So just to give you an example, like I described earlier, for this one electrode here, the principal movements of the vocal tract, tongue goes forward, tongue back goes down, the jaw goes up and down. So that movement is represented by that one particular electrode. Let's just compare that with another one here, in particular in the postcentral gyrus. And I'll go into this a little bit later, but both are important. The movements there, you can see primarily around the lip. Okay, so you have an electrode which is just millimeters away, encoding a very, very different kind of movement. So it turns out if you look at all of the different electrodes, each electrode is a column here, and each row here corresponds to different parts of the vocal tract, from the larynx, the tongue, to the lips. And these are all the different phonemes, the different consonants and vowels in English. You can see that each electrode actually has a very unique pattern. And that even as of like 5 years ago, we basically could define a dictionary, an electrical pattern that corresponded to every phoneme in the English language. There's about 40 of them. And we could define what are the spatial electrode activity patterns for every consonant and vowel. And that was exciting to think that we essentially were coming to that kind of knowledge, just understanding that we had that kind of dictionary and map. So, some of you may look at this and it kind of looks like a gene expression array, right? Which ones are going up and down. So this is really an electrode activation map for all the different sounds in English. The reason I'm bringing this up is it was really around this point that we started thinking very practically, how can we use this for the rehabilitative, the BCI neuroprosthetic application? Because if we had access to this electrical code for every phoneme, well, could we decode speech? Everything that I've been describing is what's happening at individual electrodes. What if we put it all together and try to use that information to decode speech? And the application that we and many others, including Jeff's group and others, Andrew, is to try to think about technologies that can restore function for people who are paralyzed. The one that we're working very, very intensely on is restoring speech, tapping into speech signals for people who've lost that. Stephen Hawking is one of the most famous people that where you have a huge disconnection with ALS between the cognitive facilities of the mind, but not really able to get those signals out. Later years of his life, really only able to move a small muscle in his cheek and one word, on the average of every several minutes. So we started getting into this, and this article covered some of our work actually as early as 2014. But the basic question is this: could a person who is paralyzed and unable to speak, like physicist Stephen Hawking, use a brain implant to carry on a conversation? Okay, so this is really what we've been trying to do. And one of the reasons that we're really focused on speech is because that allows us to get order of magnitude faster rate of communication. So even with typing, texting, handwriting, et cetera, where a lot of the traditional BCI work has been doing for communication, we wanted to just go straight to speech because the communication signal is so much faster and something really unique that we do. So one of the first things we did was we built an AI algorithm basically to decode speech from the brain. This was in epilepsy patients, so these are not in paralyzed patients, but we want to just do the proof of principle. And the way that this algorithm basically works is it translates brain activity into movements of the vocal tract that are in a computer. So these are simulated movements of the vocal tract, and those simulated movements are then transformed into sounds and then synthesized into something you can hear. With machine learning nowadays, you can just go end to end, meaning from the brain activity to the waveform. But one of the principles that we're really exploring with a lot of our neuroprosthetic work is trying to do things that are biomimetic, meaning that are natural. And if you understand like what information is being encoded by the brain, we should try to decode that first and foremost, because we can actually inherit and take technologies, for example, the movement to this from speech technology. So we don't want to learn all of this. We just want to understand what this mapping is. Because if you go from here to here, it requires a lot more data. Okay, it's definitely possible, but it requires a lot more data. So we're really thinking, how do we make the learning as efficient as possible? And that has to do with identifying the right representations to map to. So this is what these speech looks like. Here's the original speech that was recorded by the microphone. And here's what's decoded by the computer. It's far from perfect, but you can see a lot of the spectral and temporal energy is in the right place of the spectrogram, but still very blurry. It doesn't have all the nice contours that we find in the normal spectrogram. What was exciting to us, though, about this demonstration was that it was the first time that we actually got the sense that some of this was intelligible. So in this video, you're going to basically see the workflow. You're going to see the brain activity on the part of the brain that's controlling a computer simulation of the mouth and vocal tract. And then you're going to hear the computer generate, the speech that it was interpreting. And then you'll hear what the person actually said to compare. So first brain activity, then this computer simulation. You'll hear what the computer decodes, and then you'll hear what the person actually said. These are epilepsy patients that were not paralyzed.
The proof that you are seeking is not available in books. The proof that you are seeking is not available in books. The proof that you are seeking is not available in books. Shipbuilding is a most fascinating process. Shipbuilding is a most fascinating process. Shipbuilding is a most fascinating process.
Okay, so that's just an example of one of the more intelligible. Not great, but starting to get useful. And that was exciting to hear. So when we had that, we decided to create a clinical trial, a BCI trial. Many of you have been involved with this kind of thing. It takes a lot of work to get to the IDE, but I think because we had that knowledge and that demonstration, we wanted to move this to people who were paralyzed. So I worked with one of my colleagues in neurology, Karnesh Ganguly. So we basically created an IDE to implant subdural electrodes through a percutaneous port, covering both the arm and speech areas for people who are severely paralyzed that's impairing their communication and movement. So these are the most severe kinds of paralysis. And the first participant in this, we reported in New England Journal about 2 years ago. His name is Pancho. And Pancho had a kind of extraordinary situation. He came to the US, California, was a farm worker. And when he was 20, he was in a car accident. He walked out of the hospital next day from the car accident, but then the following day, he collapsed. He was brought back to the hospital, didn't wake up for a week after being in coma for about a week, and woke up to the realization he couldn't move his arms or legs or speak. He was locked in, and he's recovered some function since there, but he still can't really articulate speech and has very limited movements of his muscles. He lives in a care facility up in Sonoma, and he's been like that for the last 15 years. So the way he normally communicates, because he's got preserved neck movement, is he's got this stick that's attached to his Giants cap, and he types things out using this Dynavox AAC. So people who can't speak usually use these kind of assistive devices to try to get out. He can get out about 5 words per minute with that. And here's his sagittal MRI. You can see there's a lot of encephalomalacia at the level of the pons from this brainstem stroke. But the point is that all of this is working and intact. And so what we did was we basically combined an epilepsy grid to the BlackRock BCI pedestal. It's normally connected to a Utah array. And the schematic is that it goes to a computer that translates these signals and then puts it out to a screen. So there are other technologies like the BlackRock Utah array that does microelectrodes, but our focus in this particular trial was really to try to do something that was really clinically viable. And here's what the port looks like. So each one of these gold contacts corresponds to a specific location in Pancho's brain that we're recording from, and there's a HDMI cable that is taking these signals out to a computer. So the basic challenge that we're trying to overcome is translating this, the gobbledygook EEG squiggly lines from the cortical brain surface into words. And that turns out to be not really solvable 5 years ago. But because of AI, because of the development of machine learning, it is now possible. And some of the tools that we use to develop this are very similar to some of the tools that all of you are familiar with, like in Google Translate. These are sequence models that are translating from French to English sentences, for example. But it turns out these models don't really care if it's words or anything else. They're just looking for patterns of sequences that have some structure and relationship. And so in this particular case, we're translating the electrical language of the brain into actual words that we can see on a screen using machine learning algorithm. And then with this particular project, we focused on 50 words. And here's what is predicted by the computer. Here's what we asked Pancho to say. And you can see that the good news is that most of it's along this diagonal. If this was perfectly black and dark, it would mean it would be 100% decoding accuracy. But it's not 100%. Some of these areas are confusable, like it and is are very confusable. So these things that are phonetically similar are where we get some errors. And just going to show you how this works. This was 2 years ago. So someone in lab is writing to Pancho. The decoding algorithm is picking up that he's trying to speak. That's what the blinking dots means. And it's trying to figure out what word it was. And the algorithm is going to show the top ranked word, but it's in fact keeping track of all 50 word probabilities at all given times. And because it keeps track of all 50 words, you can put them in context and then do autocorrect, kind of like when you're texting, it kind of updates the spelling. So we built in an autocorrect for this as well, but it's using the probability of each of the 50 words and then projecting the highest likely one on the screen. So this next example, you'll see how the autocorrect works. It's basically an algorithm that where at that given time point, some of the word choices are incorrect. They're wrong. They're going to be displayed up here. But then it's going to use the context of the sentence to update and autocorrect the sentence into something that is grammatical and makes sense. Okay, so the context of those probabilities that you can't see are being used to project. And that's what we call language model. It's just a statistical model in English that's being used to do this. So we're using a lot of these tools from speech. And
tell me about where you're living right now.
we enrolled another participant named Anne. And Anne had a very similar story. About 18 years ago, she had a brainstem stroke. It was actually only a year after she had her second daughter. And she has severe deficit in paralysis. So really no intelligible speech. So she joined us to participate in this trial. Her story is quite incredible. But there's the encephalomalacia from her brainstem stroke there. And in this particular case, we're using a 253 channel. So we actually doubled the number of electrodes, okay, because we need more information, higher resolution, and we need still even a lot more. Here's the team. And so one of the things that we found with Anne was that when we had her decoding, for example, this NATO code word like alpha, beta, Charlie, for all the alphabets, we knew something was very different because we could get 60 to 80% accuracy within the first 2 or 3 days of training the algorithm. And then basically within about a week, she was up to about 100% accuracy on this task. And then we did something kind of unusual, which is that we froze the classifier. We didn't change it anymore. And we could continue to get very, very high performance. And this is one of the main advantages that we think about with ECOG, because it's very, very stable over time. So we could fix the algorithm and not have to change it. A lot of people use the microelectrodes have this challenge where every day the neurons are changing on the electrode array, so you have to recalibrate. This is towards something that we call plug and play, where the representations are stable. And so we decided to do something a bit more ambitious than just words on a screen. That was one of the AI algorithms that we included in this new project. But we also created a second AI engine that is decoding those neural activity patterns into sounds that you can hear. And then a third AI algorithm is actually decoding movements of the mouth. Okay, so we're not decoding text, sound, synthesis, and then also the intended movements that we're going to project on an avatar on the screen. So this is what we call multimodal speech neuroprosthetic. And there's many reasons why we decided to do this. I'm not going to get into, but here's the algorithm. There's not a lot of time. I'm happy to talk to folks after about how it works. But on the front end is a recurrent neural network and some of the language modeling that we do behind that. So here's text. So Anne can do now over 1,000 word vocabulary. We've modeled up to 30,000 words, and she can get up to about 78 words per minute with this decoder. The accuracy is still limited, so I'd say it's like 75% accurate, not 100%, but 75. And you'll see some of the errors in this video, but it is in advance because of the speed and the vocabulary increased over time significantly with more electrode density. Okay, strange sentence that got into our list here, but okay, all right, but she decoded that, fine. So here's some of the other things. So speech synthesis, for example, we have her now. So one of the cool things is that Anne could choose her own avatar. So she chose the hair color, she chose the eyes, the face, etc. Oh, and this is something that I thought was quite interesting. So we had access to about 1 hour of her wedding video. And some of the engineers we work with in lab, they basically modeled a voice based on her original voice. It's kind of interesting because her daughter thought she had a British accent. They live in Canada. It was only because the AAC device, the communication device, outputs British accent, like Stephen Hawking's. So her daughter always thought that she had a British accent. So we took her original voice, modeled it. It's not great or perfect or anything, but it is modeled after her own voice. And here's an example of how this thing works. So she'll see a prompt on the screen, and then she tries to say the same thing.
Rusty Baker.
So she sees that and then tries to say it.
I am doing well today. Anything is possible.
Okay, so I'll pause here just to show now we've got 2 HDMI cables coming out. Okay, just to give you a sense of how much data we're trying to stream out live from this. And here we're controlling a digital avatar, a fully digital version of her face and voice. And then because we built the avatar, we also, wanted to see if we can get the avatar to do other facial expressions. So here's an example of it smiling. And we're still working on the latency. Here it's very, very slow, like several seconds. But I think this is kind of opening doors to a lot of other things with digital representations of the self and more. So I think I'll probably stop here and just summarize that in this lecture, it's a really exciting time in development of what we call speech neuroprosthesis. The science is developing very fast. We know a lot more about how the pre and post central gyrus encode articulatory movements. There's this dual laryngeal motor cortex that is very novel and kind of redefines the homunculus, in a way that we just didn't understand even 100 years after it was discovered. We now know sort of how consonants and vowels are encoded by these articulatory movement trajectories in this area. And we've taken those to the task of translation and decoding. We're now in this era where it does work, and it's not just our group, there's other groups that have done this as well, using microelectrode, but also ECOG across a lot of different modalities now. And we think that this is going to be able to enhance both verbal and nonverbal communication, and optimization is going on now. So increased channel counts, better neural interfaces, better AI. So the thing that we did was extremely crude. It was just taking one of our epilepsy electrodes, and bonding it to one of these pedestals. And so you can see that there are solutions that are going to include fully implantable systems and ways to be able to do this at much, much higher bandwidth. So we're very, very excited about, how this is going. And again, I just want to conclude, and just by going back to the beginning, is that I really went down this road because of some of the work that I saw here, really inspired by the work of Dr. Ojiman, and really thinking about it. And I haven't described this work, but we're now starting to get back to this work of looking at single units, not just the ECOG, but that will be another time. But I'm very grateful to this department. And again, I'm here as an ambassador and as a friend, and really looking forward to years of collaboration with you all. Thank you.
Remarkable work, Eddie. Questions? Nina Ramirez, he runs our Neuroscience Institute at Children's.
Okay.
Okay, so Pancho, I get that the first language was Spanish.
Yes.
It uses the same string as the consonant.
Yeah.
In Spanish, but it's a modal or
Yeah. Really great question. So when this accident happened, he didn't speak English. So in fact, he's never spoken English his entire life through his own mouth. And I don't think we realized how important that was going into this. And so we're now doing a lot of redoing everything that we did in Spanish. And in fact, he's bilingual. He learned English after this accident. He's really a force of nature, really quite incredible. But one of our students is actually building a decoder that can translate whether he's trying to speak in Spanish or English. And that works, it does work pretty well, actually. It's not different brain areas, but the patterns for Spanish and English and the sequence probabilities of the sounds is specific enough that the AI can determine if he's trying to say something in Spanish versus English. Great question. Yeah. Dr. Ocean.
Many of which were from the right hemisphere, which I assume is not dominant in these patients. And I'm wondering what differences we've seen between dominant and non-dominant.
Yeah, great question. Yeah. Yeah. Right. Yeah, absolutely. Yeah. So, Dr. Ojiman asked the very important question about the laterality and how a lot of what I showed was really from the right side of physiology. The reality is that we can see it bilaterally. And something that I think is quite important lesson is that you can see the physiology bilaterally, but the causality seems to be very lateralized. So absolutely right. Some of these areas I've also removed on the non-dominant with people with having a temporary dysarthria. On the dominant hemisphere, you can get an apraxia or even aphasia if it's large enough. So part of this we don't fully understand, which is like, why it becomes so causal. And there's a lot of theories about it, but you're absolutely right that there is something special that's happening on the left side that we don't see on the right.
Wonderful work. The videos that you showed to me are like reading aloud. But what happens if the only input is spoken word? And they repeat what's been said to them?
Yes. Yeah, short answer is yes. And one of the reasons we didn't do that in this particular thing was because these areas also have auditory responses. And one of the big challenges we have right now is just making sure the decoder is not decoding what someone said to them, as opposed to what they're trying to say. And it starts to get kind of confusing. It's actually a very fundamental scientific question, actually, like, why do we have auditory processing in the motor cortex? What's its role? And it's still to be determined. One thing that we've seen, though, is that the auditory responses do shut off when we're speaking, which is good and helpful. But we don't necessarily want it decoding someone's speech from what they heard. That would kind of be cheating. It would look like it's actually decoding what they're trying to say, where it's actually just decoding what they heard. So that's one of the reasons we use spoken, but it can work for spoken as well.
So in the case of Anne, when she can phonate to some extent, obviously she can't say words on her own. What keeps her from actually phoning the sort of speech that she can make when she's sort of thinking about interacting with many people?
What's keeping her from?
So like she can phonate basically a sound, but not a word. When she's trying to say words, and when she's interacting with brain interface, she's not making those sounds.
Yeah.
How does that work?
Well, we tried it both ways. And it turns out that when she tries to speak with vocalization, it's very, very difficult and very slow. So you can think about it is that in that task, we're trying to decode activity of very, very effortful speech, because she's trying to produce the vocalization. She's trying to move the muscles in her mouth, and she can't because they're really, really weak. And so we changed the strategy. We said, we don't want you to try to vocalize. We don't want you to phonate. We just want you to just try to get the words out as quickly and as accurately as you can. So it's a different mental strategy that takes a lot less effort, because if we ask her to try to force it out through her native vocal tract, then it doesn't work, basically.
And so do they have to think about the words? And,
Yeah.
More slowly when they record that?
Yeah. Well, if she was trying to speak it, that's really slow. So that would be like that's what didn't work. Is it slower than her thoughts? Yes. And just to be clear, we're not decoding thoughts or inner speech or something. These are really the intended commands that were destined to be in the vocal tract, but never made it there, right? Because of the disconnection. And so this is an area we're trying to understand how to optimize the task, the behavior, the instructions, really to get people. I think over time it becomes more natural, intuitive, so it just gets faster and faster. She figures out a strategy in order to get these things accurate. One thing that we don't do is have her repeat the word like, I, that kind of thing. You could probably get higher accuracy, but it's not very natural to speak in that kind of way. So each of these things for Pancho and Anne, it's really just one trial for each word, and then we decode that.
Sam? It's fascinating. It seems like computational side of that. Talk a little bit about what you see the kids are in the hardware space to really make this durable answer.
Yeah. Yeah, so Anne's number one request to us, she just sent me an email last week. She's like, when is this going to be wireless? Like, she just doesn't like the idea of these two HDMIs popping out of the top of her head. So we're working on that. We just got an NIH grant to make this wireless. And I think there's a lot of parallel efforts going on in the industry between Neuralink and other companies. I think it's going to happen, I think, in the next 5 years.
Randy, this is a very esoteric question. A long time ago in cognitive psychology, there was a big argument between B.F. Skinner and Noam Chomsky about cognition. B.F. Skinner thought it was mechanistic. Chomsky essentially thought it was not mechanistic. And one of the major experiments in that was to put someone in a neuromuscular blockade and then have them perform a cognitive activity. And when the NMD was reversed, they could show that they had processed this, which was a strong argument against B.F. Skinner, the mechanistic thing. You've taken this to a whole new level. Any thoughts perhaps on what this means, not just the sort of speech, but and verbalization, but actually the cognition?
Yeah,
It's actually
yeah. Yeah, that is really more on the philosophical side. I would say we're not there yet. We'd like to get there, because I think the speech problem is starting to look not completely solved, but we're getting there. The cognition is really, I think, the big, more interesting question, and I think that that's where, the rest of my life, I think we'll be thinking very hard about those kind of questions. What is the inner speech? Where is the planning for these structures coming from? And, expanding out from the motor and sensory areas to these more cognitive. And we're very interested in those theories because they're very fundamental.
Yeah, it's actually very similar question. Just in the last one, expressions. Just another esoteric reference to where you're asking surprise for just how far have you taken just the simulation of the facial muscle unit? Because things like micro expressions or other cues from facial things that are supposed to make is that something?
Yeah. Yeah. Well, so the question is really like, how far can we go with some of this expression decoding? And one of the things you notice is this avatar is like making kind of natural movements, not just like looking still. And those are not decoded. Those are actually like built in. So one of the things, and this is a general thing, but I think why right now is such a special time for this kind of work is because of generative AI that's coming online. And so one of the challenges that we had with decoding before was that you really had to be 100% or it didn't work barely at all. But because generative AI can fill in a lot of the holes, it's predictive, it is complete in many ways. You just need to get enough information to the decoder to get it, started. So that's why I think we're very excited. Like in the case of an avatar that's moving, you don't need to decode every precise, joint movement. You just need to know the general picture, the general velocity is a command, and the generative AI will essentially fill in all the details. So that's, it could do it wrong, but I think that this is going to become very, very natural looking, and interactions, let's say, one of the things that we're thinking about with Avatar is how people will interact in digital spaces like the metaverse. So some person will be paralyzed in the future and on the metaverse, but actually moving around and speaking and interacting with other people who are not paralyzed, but in that kind of virtual form. I think there's a lot to push in terms of how much better we can get the decoding.
Follow.
Yeah.
I mean, obviously that makes this work, but the generative AI capability. I just wonder what sort of safeguard or how have we wait back when we are doing this? We're actually kind of, especially in the case where we may not have a gold standard for that processing more. How are we trying to keep from like making mistakes?
Yeah. Well, it's interesting. So the question is really about safeguards and maybe mental privacy. And what you saw was basically Anne projecting volitional, probably not emotion-based expressions, right? I asked her to smile, asked her to look surprised, etc. If you really wanted to drive a naturalistic facial expression decoder, you could probably put the electrodes in a different place, like the amygdala, some of these other areas that are processing emotional affect, right? And I think that is something that would be very interesting, which is having the avatar express what the inner emotions, if that's what you actually want to do, right? If that's what you actually want to do. I think everything here that I've shown is very volitional, and it's very related to the intentional volitional control of movements. But there is a very, very serious question, an important question about where this crosses over the line in terms of mental privacy. Like, could we in the future decode what someone is thinking about? And I don't see why not, right now. I don't think we're there yet, but I don't see any reason why not. As the technology scales, we have to be thinking about those really important questions, sooner than later.
Christoph, you get the last question.
Yeah, very good question. I find fascinating. We have these signals from the cortex, we decode them, and in the end of the day, now you have a real neural network that you're analyzing. So is any backwards engineering from your engineers and sort of seeing new patterns that the cortex uses that we don't use right now for modern computing and AI generation? So in other words, you're reverse engineering from the real neural network back. You're like, wow, this is how the cortex does it. Maybe we can do the same thing with our computer.
Yeah, really fascinating question. That's like a dream if we can do that. If we can take AI and make it super AI, based on how our brain works. And we're not there yet in the work that we do. We're very applied, in how we use this right now. But we are thinking, we're actually directly comparing how an AI learns language, for example, with the neurons that we record from some of these speech areas. And some of it actually looks very similar, but some of it looks very, very different as well. And so the similar parts, I think, are understandable because the AI is learning the statistics of language and optimizing around it. And so is our brain. So it's cool to see that, but it's not surprising. one of the areas that we're really focusing on is where the differences are, like how the human brain neurons are computing different than the neurons in the neural network, and we see those differences, but we don't understand them yet. So hopefully that will give us an idea how to make these things, you know, maybe smarter and more efficient, but we'll see. Yeah, great question.
Thank you, everybody. Give Professor Chang a hand.